Okay.
That's music's fine.
Music's fine.
Music's fine.
Wait, no, but you gotta hear them.
Volume.
You'll hear us soon.
I can hear you.
Yeah, you guys were just muted, so you can unmute if you want to say something. Just still people are coming in. You all have your videos. There's me. Okay. There's more people. Look at this. Everyone's joining. This is great. Okay, we're gonna go and get started in just a couple minutes. I'm gonna mark off who is here. Amy Barcel. Okay, do it another way. Oh, the sun is coming in. Looks like we have almost everybody here. So what I'm going to do is try to take attendance when, sorry, this light, I'm trying to, I have lighting issues in this room. Is that gonna work? Well, you're gonna be looking at the PowerPoint soon anyways. Okay, I'm going to just call attendance. Unmute yourself when you hear me call your name. Let me know you're here so that I can get your attendance in, okay?
First, hang on.
Is there a question? No? Okay. Amy Barcel. I see you.
Here.
Thank
Here.
you. Danielle Barkhurst.
Here.
Thank you. Eric Bennett.
Here.
I see your name. Okay. Kirsten Brutus.
Here.
Yeah. Okay. Kira Castle.
Here.
Maria Collins.
Here.
Okay. Ashley Conley.
She's here. Her audio is just not working.
Audio's not working. Let's see if I see her. Okay. Grayson Esnick. As Sink. Here. Okay. Crystal Fortune.
You go ahead, it's Bill's already talked to us. So.
Okay. Jason Hansen.
Here.
Okay, thank you. Madeline Johnson. Okay. Put a question mark there. Ryan Kronberg.
Go Big Red.
Go Big Red. Okay. Lindsay Kritsky. I can't.
Here.
Sorry, I messed your last name up. Macy Lawler. Okay, question mark. Corinne Longo.
Here.
Okay. Jessica Lucero. Okay, question mark. Elizabeth Morales. Okay. Anna Moran.
Here.
Okay. Morgan Reef.
Here.
I already got Anjanette. Olivia Rogers. Still trying to figure out if I can make this lighting better. Oh, it's kind of better. Is Olivia Rogers here? No? Okay. Maze Shalau. Okay. All right, we did pretty good getting everybody on here for the first Zoom meeting, which I'm sorry that we have to do it this way, but we have to be super careful. I get to go and get my COVID tested tomorrow. I'm so excited about that, but do that just to be on the safe side so that we all are safe and know if we need to worry about anything. So I'm gonna go get tested just so that we can put our minds at ease. Okay, I am looking at pulling up your lecture manual. We will be looking at it a lot, or you will be needing to study it a lot, especially when we start getting into the skeletal system. And hopefully we get into the skull today, and there is going to be a lot to learn. So just kind of a heads up about that. And I will try to point out as many features as possible. This is where your lab is going to be really helpful. Get your hands on skulls, look at those features, look at those different bones. It's all going to make more sense when you can actually touch it and see it, more than just a picture. Okay. So we are working on cells and tissues. So I'm going to pull that up. If I remember how to do this. See, I think I have to share my screen. There it is. Okay, can you see the PowerPoint?
Yeah.
Yeah, okay, awesome. Now, oh, there's 2 more people here. Let's see who they are. Elizabeth Morales and Macy Lawler.
You forgot to mark me down as well.
And what's your name?
Chelsea Gomez.
Chelsea Gomez. I'm looking for your name right now. I don't have you on this. This list is a couple. Oh, Chelsea, I have you down. You're checked.
Oh, okay.
I think I saw your name on the side and just checked you off. Okay. All right. I should probably let these people in. Oh, who's Megan? We have another person coming in. Megan. I'll ask. Megan, what's your last name? She's on her cell phone. Sorry, I'm just trying to get our attendance squared away. And I want to make sure Megan gets credit for being here today. Okay. All right. Now, quick question. Does anybody remember where we stopped off on cells and tissues? Because I'm honestly, when I was looking through this yesterday, couldn't remember where we stopped. I know we were talking about the different types of epithelial cells.
That was it.
Right there or right here?
About right there.
Okay, perfect. Thank you.
We hadn't got to stratified squamous. I know that.
We did talk about stratified squamous? We did not.
No.
Okay.
No, we didn't. We only got like 2 PowerPoints in.
Okay. So let's see. We'll just start at epithelial tissue again. So we taught there's 4 main tissue types. So we have epithelial tissue, connective tissue, muscle tissue, and nervous tissue or neural tissue. Those are the 4 main types. So the first type we're going to talk about is epithelial tissue and talk about how we classify it. So first of all, singular is epithelium. Plural, is epithelia. So if you're talking about more than one cell, it's epithelia. And I have to catch myself because I will get those confused myself sometimes. Okay, classes of epithelia is based on the shape of the cell. So squamous epithelia is that thin, flat, kind of pancake-like cell. Cuboidal epithelia is square-shaped or kind of like rounded square-shaped, more like that. They're not going to be perfect, but what you do find is that the nuclei are always lined up kind of nice in order like that. Columnar epithelia, how can I erase that? Oh, sorry. I'm still playing with this computer, so forgive me. Columnar epithelia, those are thin, tall cells, and we have a picture of those coming up. So we name epithelial cells based on their shape and on how many layers there are. So simple refers to 1 layer. So simple epithelium is a single layer of cells. Stratified epithelium is more than 1 layer of cells. Okay, so looking at just the basic cell types, we have squamous epithelium, thin pancake cells, and they're always going to be pretty thin. Simple cuboidal, note that the nuclei are all kind of lined up. That's kind of one of the features that you'll see with simple cuboidal. And then simple columnar are tall, skinny, column-shaped cells, and the nuclei are mostly lined up in this type of cell as well. And it's common that you will find that the apical surface, a good term for you to know, the apical surface has cilia. You'll find that very often. And so, for example, you would find simple columnar epithelial cells in the lining of your respiratory tract, and those help move up mucus up and out of your respiratory system. So they have a really important job, and that's what the cilia helps to do. Okay, trying to move on to our next slide. Something just happened. Bear with me. Okay, there we go. Okay, so more than one layer. We have stratified squamous epithelium, and you'll note that kind of down at the bottom, they're more cuboidal in shape, but the closer you get to the outer surface or the apical surface, I'll write that nicer, apical, and then down here is your basal layer. And you have your basement membrane, which is one of those features that you find with epithelial cells. So the closer you get to the apical surface, they are more flattened out. And if this surface is, for example, on our skin, those cells dry out, keratinize, and begin to act as that waterproofing layer. Okay, stratified cuboidal. Very rarely would you find it more than 2 cells thick. Very rarely are you going to find it more than 2 cells thick. And I'll show you some pictures coming up, some real pictures of these. And then stratified columnar, what you have is instead you have cuboidal layers on the bottom, and then the uppermost apical right, layer is going to be your columnar cell, okay? But this gives you multiple layers, but it's called stratified columnar because you have that columnar layer. Okay, here are some real pictures of squamous epithelial cells. One place that you would find these is creating the alveoli in the lung, which is the little sacs that inflate with air and are surrounded by blood vessels. So this is where gas exchange actually occurs. So they're involved in absorption of gases and secretion of gases. So absorbing oxygen and secreting carbon dioxide, for example, the main job there. And so there are these little sacs that almost look like bunches of grapes, and they're made of a single layer of squamous cells, and then they're thin and flat, which makes it much easier for substances to absorb through the cell into the bloodstream or out of the bloodstream and back into the lungs. So kind of an important function of those cells. Keep hitting the wrong button to move forward. Sorry. Okay, here is a picture out of a, oh, that's much better. Picture out of a microscope of stratified squamous epithelial cells. And it's kind of hard to see the individual cells, but you can see the nuclei. And they're packed pretty tightly, and you get closer to the surface, you can tell that they're kind of flattened out. Okay? And so we would find this type of tissue on the skin, your mouth, your esophagus, other mucous membranes. These are for physical protection. So you have these extra layers that are able to slough off without you damaging that tissue. So having those extra layers is protection. Okay, another picture of stratified squamous epithelium. So places you would find this, other places, remember all of the mucous membranes are going to have stratified squamous epithelium. So lining the surface of the skin, lining the mouth, the throat, the esophagus, the rectum, the anus, and the vagina will all have stratified squamous epithelium. And many layers allows cells to slough off without causing damage to the underlying tissue. So one reason why you want to have a lot of layers, and a lot of layers also protects the underlying tissue from physical attack from bacteria and viruses and abrasions, etc. We have another person coming in. Okay, simple cuboidal. This is a microscope picture of the nephron loops in the kidneys. And so they are simple cuboidal little loops. And so if you look closely, you can see they're almost like little circles. You can see the nuclei. Okay. They're square or kind of square roundish shape. Find them in glands, so glands that secrete things, urinary tubes, the kidney tubes. So this is the kidney tubules in this picture here. And they function in secretion and in absorption. So we don't get to talk about that very much in this class since we're doing just anatomy. I saw that. But in my physiology class, we are definitely going to talk about how these work in secretion and absorption, and that's going to be fun for them. If some of you are in also physiology, you have that to look forward to. Okay, here is a closer up picture of simple cuboidal epithelium. And this is a great shot because you can see those cells. They're kind of roundish squares. You have those nuclei all nice and neatly lined up in a row. See that? Woo! That's one of those things that you can expect to see with simple cuboidal epithelium. So for our next quiz or exam, which I have our schedule, let me pull that up really quick. I just folded it in here in a weird place. Okay, here it is. Okay. So not until our exam will you be tested on the cells. There will be pictures of cell types. I will make them very clear, try to make really obvious pictures that you will need to be able to identify the cell type. Okay. So cell types that we've gone over in class would be cell types that you would need to be able to identify. So I'm trying to point out identifying features for you. So to make sure you're taking notes on that. And again, I am recording these videos and I have to save them and then I have to upload them, which takes a long time, but they'll eventually be onto YouTube and I'll put a link on our Canvas page that you can go back and watch these again. So hopefully that will be helpful for you. That will usually take me several hours before it's up and available. So since we're lecturing at night, it might not be available till tomorrow morning sometime. So just as an FYI about that. Okay, stratified cuboidal epithelium. So this is a little more messy. You note that the nuclei are not all nice and neat in a row. So if we're looking at the actual picture here, we have a tubule. And the outer layer, you can see the nuclei are in a nice row. Okay, so the outer layer is pretty good, but then when we look at the innermost layer, we have these nuclei kind of randomly scattered. And so it gets a little messy when you add a second layer to cuboidal epithelium. So that is one of those features that you'll see. Is you can, in the drawing, you can see that too, is that you don't get to see all the nuclei in that second layer, which makes it look kind of random, a little messy. But your outermost layer is always going to be nice and neat. Oh, I'm sorry. Let me back up real quick. Where you would find stratified cuboidal epithelium, ducts of sweat glands. And these function to secrete and absorb materials, so secreting sweat, secreting other materials, and absorption. Okay, this is a great picture of simple columnar epithelium. You can see how nice and neat those nuclei are all lined up right down the middle. It's beautiful. Nice layer. You can see the cell walls. This one's a pretty easy one to identify. Okay, column shaped. Places you'd find this: your trachea, small intestine, uterine tubes, and they also function in secretion and absorption. And again, how they function in the body is a physiology topic. So I'm trying to restrain myself right now. Okay. Now, here's another picture of simple columnar epithelium, a drawing of it, and this one is showing that it is ciliated. So if it is ciliated, if it's a drawing and you can tell that it's ciliated, you must also state ciliated simple columnar epithelium, because that is the more specific answer to what kind of cell type it is. In this picture here, I didn't notice, but these are microvilli. So places you'd find it with microvilli would be in your intestines. And those microvilli actually act to increase the surface area so they can increase the amount of absorption of nutrients from your intestines into your bloodstream, which again is a little bit of physiology, but you'll survive. You'll survive. So places you find it lining the stomach, intestines, gallbladder, uterine tubes, collecting ducts, and kidneys. And you can find these in many different places. They act as a protection, secretion, and absorption. So specifically in the intestines, these are great for protection and absorption because you have these microvilli that act to increase the surface area so you can absorb more nutrients. But then look how long this cell is. It takes a long time for substances to move all the way through the cell into the underlying layer, into the bloodstream. So that is actually a function of protection that helps protect your bloodstream from things that shouldn't get in there. Having these long cells makes it harder for substances to move all the way through them down to the base into the bloodstream. Okay, stratified columnar epithelium. Some of the places that you would find this in the pharynx. So the pharynx, your pharynx is kind of back in this area here in the back of your throat. Okay. The epiglottis, which is down here, is that portion that kind of folds over when you swallow to protect your windpipe so that substances go down your esophagus and not your windpipe. The anus, mammary glands, salivary glands, and the urethra all are lined with stratified columnar epithelium. And their job is protection. So remember, the thicker a cell is, the more protection it offers in the sense of having substances pass through them is going to be more difficult. it's going to be really easy for a substance to pass through a squamous cell because it's so thin. But the thicker a cell is, the harder it is for substances to pass through. Okay, pseudostratified ciliated columnar epithelium. This is a fun one. And this is one I promise I will not have you identify because it's tricky. It has the appearance of other cell types. It really truly is one cell thick, but these cells are so squished together is that the nuclei squeeze up and squeeze down. They're not lined up. And so they're just jumbled together, even though they are just columnar cells, they're jumbled together and nuclei are squeezed up one way or squeezed down another. So it looks like you have multiple layers of cells where you really only have one layer of cells. That's why it's called pseudostratified. Pseudo not meaning it's not real. Okay, it's ciliated, so the cilia at the apical surface, and you will find these lining the nasal cavity, the trachea, the bronchi, parts of the male reproductive tract. And they work with mucus production, secretion, and moving the mucus. So those ciliated cells move the mucus up and out of your respiratory tract so that you swallow it or cough it up, gross, to get it out of your respiratory tract, which, you know, we have COVID going around right now, and that's going to cause those cells to work in overdrive, and they're not going to be able to keep up with people who were severely sick. Okay, so remember this. One layer of cells, the nuclei are at different heights. They're all kind of squeezed up and squeezed down and jumbled around. So it looks like you have multiple layers, but you really do not. That's why I'm not going to ask you to identify this cell type because it is a tricky one. You will find that pseudostratified epithelial cells are ciliated. And so, for example, portions of the male reproductive tract, like lining the vas deferens or the seminiferous tubules, that would actually help move the developing sperm along as it continues to develop in the testes and the epididymis. So they have different purposes, but the cilia is helping with movement. Okay. Columnar. Transitional epithelium. This is another one I typically don't ask you to identify because it's a little harder to do. What is cool about transitional epithelium is that it has the ability to tolerate cycles of stretching and relaxing, stretching and recoiling, and returning to its shape without damage. So the major place that you would find transitional epithelium is the bladder. Think about it. If you have to go to the bathroom really bad, your bladder has stretched out. After you've relieved yourself, it has recoiled to a much smaller shape. So, because it stretches and recoils, the appearance of the cells change depending on whether they're stretched out or they're relaxed. So we find this, I want to say, only in the urinary system. So the bladder is the big one, but you do also find it in the ureters. I'm going to stop there and not say anything wrong. Okay, so the type of cells that you would find making transitional epithelium would be squamous or cuboidal. So here we have some pictures, some examples. And here is relaxed epithelium, so a relaxed bladder. You have the cells, they almost look like they're jumbled on top of each other. They're nice and fat and round and happy. But what's really cool about these cells is that when your bladder is filling up with urine, these cells have the ability to kind of stretch out. And they're all stretching out together, and that thins the bladder wall, but allows it to expand. And so you go from having an empty bladder with kind of rounded, happy cells to a stretched out bladder where those cells are more stretched out, more compact. And that is what it's going to look like when it's in a stretched out position. And then when you relieve yourself, that bladder recoils and those cells recoil again back to their original position. So they're really cool. They don't slide past each other, but they stretch out, which is a really cool thing that they can do. Okay, muscular tissue. This is skeletal muscle. I had to search to find one I thought was good enough for us, or one that we could really see what we wanted to see. One of the big features of skeletal muscle is that it is striated. And these lines that you see, those are the striations, okay? These skeletal muscle cells are very long. They are multinucleated, and that is a function of development. And early in development, those cells were all single cells and had a single nucleus. But as you mature, those cells in development grow together but keep the nuclei. So you end up getting these long, thin cells with multiple nuclei in them. This is one of the few cell types you're going to find that is multinucleated. So these dark dots that you see in the picture, these are all nuclei, okay? Skeletal muscle is responsible for voluntary movement. So you control that most of the time, unless you're getting a twitch or something. Voluntary movement, so for example, arm and leg muscles. Here's another illustration of skeletal muscle showing you, this is a great one because it shows you the striations really well, the multiple nuclei, and then kind of the shape, long, thin, almost, well, they're kind of like tubes, long tube-like shapes of these cells. And then they're all bundled together. Okay, this is cardiac muscle. And cardiac muscle has some different features. You can call them cardiocytes or myocytes. So some of the things that you're going to find are different. They're branched. The cells branch. They branch and join up with each other and branch and join up with each other. So they're not straight lines like we saw with the skeletal muscle. Another feature is these lines you see here that I'm circling. These are intercalated discs. This is the place where the cardiocytes come together, or where they have their intercellular connections between one cell and another cell. And they do have intercellular connections because the cells communicate with each other in the heart. And you will only find these types of cells in the heart. And they also have the ability to create their own rhythm, their own sinus rhythm, which is really cool. And we won't talk about that in this class again. He keeps putting his thumb up. Thank you, Ryan. So here's another picture of cardiac muscle. So things to remember, and I don't like that they didn't put that they're branched. They are branched, and that is one of the key features of cardiac muscle, is that they are branched and that they are joined together at these spots, these dark lines that are your intercalated discs. These are not multinucleated, okay? These ones are just one nucleus per cell. So the skeletal muscle and the multinucleation is unique. Okay, moving on to smooth muscle. And one of the things about smooth muscle is when you look at it, it does look smooth. And it has that appearance because it lacks striations, no stripes, okay? The cell itself kind of is spindle shaped. So let's see if I can draw this here. Kind of spindle shaped like that with the nucleus in the middle. And they kind of all fit together. Drawing another one, okay? And smooth muscle is actually the strongest of the 3 muscle types. It has the ability to hold a contraction for a long period of time. So think about, for example, contractions in childbirth, sorry, childbirth. So one of the places you would find this is in the uterus. The muscular layer of the uterus is made up of smooth muscle. You also have smooth muscle lining the blood vessels. Sphincters, so the urinary sphincters or anal sphincters, you have 2 of each, and one of them of each is made of smooth muscle. The other is of skeletal muscle. So you have one sphincter that's voluntary and one that's involuntary. Smooth muscle is involuntary. You cannot control when that muscle contracts. That is controlled by your nervous system. You cannot voluntarily make it happen. Here's another great picture of smooth muscle. And it, again, when I look at it, I just see smooth looking cells. Lacking the striations, they're just kind of streamlined little cells with one nucleus per cell. Okay, I'm gonna grab a drink and we're gonna go on to neural tissue. Okay. Neurons. Okay, so this picture here, you are looking at 2 neurons. The 2 large cells there are the neurons. And then they have multiple extensions off of their body. Soma is the body of the cell. Then you have the dendrite and the axon. And for these cells, it's kind of hard to tell in the picture which is the dendrite and which is the axon. Just because this is a real cell, you can have multiple dendrites. You can have one axon, but it's hard to identify it. We'll have an easier picture to come up for that. Places you would find this, also note a nice obvious nucleus in the center. And you have these branches that come off. So multiple branches. So it's very common to have multiple dendrites. Usually you have one axon. That axon can branch and can do different things, but typically you have one axon. Okay, brain tissue and nerves. This is where you find neurons. And the main job of a neuron is to conduct impulses, send signals. Receptors receive a signal. They need to send that signal to the control center. It's being sent via neurons. That control center is made up of neurons that process the signal and then decide what to do about that impulse and send a signal back out to the effectors using neurons, and then causes the effectors to act. So it's conducting little electrical impulses while it's sending signals. That's how it sends signals. Okay, so here is a simple schematic of a neuron where you have all these little dendrites, okay? They look like a tree, and one long axon. And these dendrites, each little spot here, each one has the ability to connect to another neuron. So one neuron can have connections with hundreds of others via these dendrites, which is really cool. Then you have the cell body, your nucleus, your internal organelles of the cell, the nucleus and the nucleolus, which we're going to talk, when are we going to talk about the cell? We may not be talking about the cell. Maybe that's physiology. No. We're not going to talk too much about it. Yeah. He's happy about that too. Yeah, I think we'll get into it more in physiology. Okay, then you have this one long axon that ends in these synaptic nodes, and they connect to other cells or neurons via these synapses. There are these little gaps between the synapse and the next cell where little neurotransmitters travel across and bind to receptors and cause the signal to continue to travel on. Sorry, that was a little bit of physiology. Here is another picture of a neuron. You can see the cell body here pretty well. See these extensions coming off are dendrites. They have been able to determine that this extension is the axon, so I'm going to believe them because it's hard for me to tell the difference in a real picture between an axon and a dendrite, if I'm going to be honest. You have your nucleus and the nucleolus. The nucleolus is the most darkest portion of the nucleus. And then surrounding a neuron, you're going to find little tiny nuclei. And these nuclei are from special neurons called neuroglial cells. And these are essentially helper neurons. And they help support and nourish the main neurons of the nervous system. Okay, so this is the same picture we were looking at before, but now we're focusing on the glial cells. And you can call them glial cells or neuroglial. Either one is perfectly fine. Usually I just say glial cells, just I guess because it's shorter. But so you see these little dots, these are the nuclei of those supporting glial cells. They're everywhere, okay? They surround the neuron. They function for protection, for nourishment, support, insulation. They all have different functions. Okay, so this I pulled from the internet so you could see the different types of glial cells that are found in the nervous system. And they're broken down based on the central nervous system and the peripheral nervous system. I do want you to know this slide. So I expect you to know the names of the different glial cells, where they're found, and what their major function is. So for example, if we look over here at astrocytes, I like these because, well, they're called astrocytes, so they're kind of star-shaped cells, and they have these extensions at the ends that wrap around the neurons. And they wrap around blood vessels. You find them in the central nervous system, and they wrap around blood vessels and create an extra layer around the blood vessel, which forms the blood-brain barrier. And that makes it much more difficult for things from the bloodstream to pass into the central nervous system. So it is extra layer of protection for the central nervous system. So that's one great example of a glial cell. You find it in the central nervous system, and it's helpful in the blood-brain barrier. That's kind of what I would ask you. Oligodendrocytes. Oligodendrocytes and Schwann cells are very similar. The main difference is they're found in different places. Oligodendrocytes are found in the central nervous system, and they are involved in myelinating axons. So they basically wrap themselves around and around and around an axon, and it creates this insulating layer, which makes it easier for signals to pass faster. So having an axon myelinated is really beneficial. And so because of that, we have another cell type in the peripheral nervous system that does the same thing, that myelinates axons. And in the peripheral nervous system, that is the Schwann cells. So they wrap around the axon and do the same thing. So those are a couple that off the bat are really important to know where you find them, what they do, okay? Let's see. What do I want you to know about satellite cells? Satellite cells are really cool, actually. These guys, and they didn't put the function that I think is cool about them, but they wrap themselves around the body of a nucleus or around a knot of white matter, and it protects it. And it can protect it from things like heavy metals like mercury or lead. It adds this insulating protection around it, which they didn't put down there, but that is one of the things that they do. They can also, it says, regulate oxygen, carbon dioxide, nutrient, and neurotransmitter levels. So they wrap themselves around a group of neurons to protect them. Ependymal cells up here, these guys line the ventricles of the brain, which we'll talk about later because we'll be talking about the brain. But inside your brain, you have basically 4 cavities filled with cerebrospinal fluid. So lining those cavities are these ependymal cells. They help circulate the fluid. They help produce that fluid, and that fluid provides nourishment to the neurons. So it's a very important function. And then lastly, microglial cells. These guys are like little trash cans, little trash compactors. They go around finding finding debris, waste, cellular debris, pathogens, things that do not belong in the nervous system, and then engulf them by phagocytosis to get rid of them. Okay, so that's the quick version of what cells there are, where you find them, and their main job. Okay, now we're going to move to connective tissue. This is kind of a busy slide, and I thought long and hard about how can I fix this, and it was going to take a long time. So we're talking about connective tissue. There are 3 main types of connective tissue, okay? So we have connective tissue proper, supporting connective tissue, and then fluid connective tissue. So let's just start with connective tissue proper first, and then we'll move on to the other ones, and then we'll look at some pictures, okay? So with connective tissue proper, there are 2 main types of connective tissue: loose connective tissue and dense connective tissue. So I could draw little tiny arrows, kind of breaks it down a little bit. What they have in common is that the matrix, or the substance that they secrete, is fibrous. So connective tissue is a product of the cells. It's not the cells themselves. And you'll see this when we look at the pictures, okay? But connective tissue proper is made up of mostly fibers that the cells produce. So examples of, loose connective tissue, we have loose areolar connective tissue, adipose tissue, and reticular tissue. Over on the dense connective tissue side, we have dense regular connective tissue and dense irregular connective tissue. And so we'll look at pictures of all of these and see what the difference is of these and how can we identify them. And then we look at the supporting connective tissue, number 2. You have either a gel matrix, so cells secrete a gel substance. That's the tissue itself, is this gel substance that the cells secreted. And that is mostly known as cartilage. There's different types of cartilage, but it is mostly a gel matrix with some cells interspersed. And then also a solid matrix, which a good example would be bone. Okay, lastly, there is fluid connective tissue, and it has a liquid matrix. And so the best example of this is blood. It's you have blood and components dissolved in a fluid that is considered a connective tissue because it's secreted by the cells. So some people have a hard time wrapping their heads around the fact that blood is a connective tissue, but it truly is. Okay, this we are looking at loose areolar connective tissue, or just areolar, but I like to say the whole thing out. The type of cell, so you see the nuclei, the dark circles, those are the fibroblasts. These are the cells that make all those little fibers you see, those little lines in the picture, those are made by the fibroblasts. And so those hair-like fibers are called fibrocytes. And what their function is, one of the functions is it helps attach tissue to other tissue. So for example, attaches your skin to the underlying muscle, or muscle to muscle. When you get to dissection, you'll see that when you're kind of separating muscles, it almost looks like white spider webs in the tissue that you're separating. That's the loose connective tissue, the loose areolar connective tissue. Those fibers are those white spider webby appearing structures in the tissue. But they can be very strong and help hold tissues together. Okay, here is a closer up picture of loose areolar connective tissue. You can see that the fibrocytes are the small dudes. There are macrophages, which is a type of white blood cell, and macrophages are found in the tissues. They kind of wander around tissues looking for things that don't belong, bacteria, virus infected cells, pathogens, etc., and engulf them. So you'll find macrophages in all sorts of different tissues. Mast cells, which are interesting, these are also another type of white blood cell that secrete histamine. So they're responsible for your allergic response. If anybody's been having allergies lately due to the high pollen content. Yes, me too. Okay, and then you also have these elastic fibers. There's 2 main types of fibers. The thin lined ones, those are your elastic fibers, and the thicker pink ones right here, those are your collagen fibers. So in the loose areolar connective tissue, you have elastic fibers and collagen fibers. I'm not going to ask you to identify the fibers. I just want you to identify the tissue type, okay? So again, I'm going to try to make sure I use the clearest possible pictures for you when you are being tested on this. Okay, Anybody want to tell me what the cell type is right here?
Columnar.
Yes, that's right. Is it simple or stratified?
Simple.
Simple.
Excellent, excellent, excellent. Okay, so then looking below that simple columnar epithelium, you see these round cells? They're kind of globular shaped. That's our hint. These are adipose cells. Adipose cells tend to be round. They're full of lipid droplets. The nuclei is typically squeezed out to the side because it's so full of lipids. You will find this under the skin, or more properly, let's say it's deep to the skin. So under our skin, we typically have a layer of adipose. Surrounding our internal organs, we have adipose tissue. The breasts are primarily made up of a lot of adipose tissue. So those are some examples of where you would find it. Functions of it: insulation, padding, cushioning, temperature regulation. Those are some of the functions of adipose. So I always tell my students that adipose is not a bad thing. It has very important functions in your body, and you need to have some. So just keep that in mind. Okay. In this picture here on the right, you can actually see where the nuclei are squeezed out to the side. This big fat cell, there's the nuclei, and then there's the rest of the cell full of lipids. So they are storage molecules. That's what they do. Okay, this is reticular fibers or reticular connective tissue. And the easiest way to identify reticular connective tissue is to look for these dark branching fibers. Okay, and they're branching, they're dark. You don't see this type of fiber in any other cell type. They're secreted by fibroblasts. Those dark fibers is characteristic of reticular connective tissue, and it is important in the framework of many organs: liver, spleen, appendix, tonsil, lymph nodes. In fact, in the lymph nodes, it produces the little chambers where lymphocytes hang out ready to fight infections. So they have, it helps form kind of the septums or the little walls inside lymph nodes for that purpose. Here is another picture. This is a good one, where you can clearly see those dark branching reticular fibers. You can see the little reddish cells interspersed between there, but those dark branching fibers is what you want to look for when we're looking at reticular connective tissue. Okay. This is dense connective tissue, and the matrix is secreted by fibroblasts again. The fibers line themselves up, usually parallel to each other. So this picture here is great because you can see they're almost straight lines of parallel fibers, okay? Places you would find this dense, and just so we know, it's dense, regular connective tissue, okay? It's dense, regular because it's those parallel lines. We also have dense, irregular, and it looks very, very different, okay? But where we find this dense, regular connective tissue, your ligaments, your tendons, these are bundles of collagen fibers, okay? When you look at them, they're pure white. There's not a blood supply in those suckers. They are there for strength, okay? So tendons, ligaments, and aponeuroses, which are connections between muscles, like sheets of white connective tissue. This is also dense, regular connective tissue. Note that they're still parallel, but they're wavy. Okay, still parallel, but they're wavy. So still dense, regular connective tissue. And it is made up primarily, as I mentioned before, of collagen fibers, which you want to remember. And you can see those fibroblasts are responsible for secreting the collagen fibers that make up the tissue. Okay, a little bit about dense, regular connective tissue. It does heal very slowly after an injury, and this is because there's very little blood supply to the tissue itself. And that is to help increase the strength of that tissue, but because there is very little blood supply, it takes longer to get the nutrients and the materials that are needed to repair a damaged tendon or ligament or aponeurosis. Okay, main types of dense tissue, you have tendons. Tendons connect muscle to bone. And you need to know this, the difference between tendons and ligaments. Ligaments connect bone to bone. So both of those play a very important role. They're both made of dense, regular connective tissue. Aponeurosis connects muscle to muscle. Okay? So this picture here is actually a drawing, but that's okay. This is a drawing of dense, irregular connective tissue. And if you look at it, it doesn't look anything like the dense, regular connective tissue, which was parallel or kind of wavy. This is kind of all over the place. It's a jumbled mess. But this is a connective tissue. Where we would find these, capsules surrounding internal organs, nerve and muscle sheaths, the dermis layer of the skin. So you have 3 layers to the skin. You have the epidermis, the dermis, and the hypodermis. So the middle layer, the dermis, is where you find this dense, irregular connective tissue. So functions, it helps prevent or resist changes in directions. So if you were, because it's in your skin doesn't want to twist and turn and do all sorts of things. It's going to hold its shape pretty well. Well, that's because the dense, irregular connective tissue in there resists forces and changes in directions. It helps prevent overexpansion of organs. For example, the bladder, you wouldn't want it to overexpand. So there is a capsule surrounding the bladder that kind of caps it off and says, okay, you're full. It's time to empty. Okay, fluid connective tissue. We are looking at blood. So, I don't know what the dark, okay. The blue arrows are pointing to these dark dots. These dark dots are white blood cells, okay? The red little circles around them, those are red blood cells. So the types of cells that you would find in blood, you have erythrocytes, which are your red blood cells. You have your leukocytes, which are your white blood cells. And then you have thrombocytes. And I'm going to have to review what thrombocytes do. Forgive me. Let me double check real quick.
Clotting.
Clotting?
Platelets.
What was it?
They're kind of like your
Platelets.
Platelets.
Oh, okay. So that's just a
Platelets.
different word than I'm used to. Okay. Yes, I do know my platelets. Okay. Excellent. Because you guys did your homework already. Okay. So we find so we find this in your circulatory system, and that's pretty much the only place you're going to find it is in blood vessels, whether they're all the way tiny vessels, capillaries, or very largest vessels in your body like the aorta. That is where you're going to find them. White blood cells can be found in other tissues. It just depends on what type of white blood cell we're talking about. But I don't believe we're going to get super detailed about white blood cells. Okay. 3 main cells that make up blood. We're doing pretty good. We got 15 more minutes left for today. Okay, 3 cells that make up blood is erythrocytes. Should know that these are the red blood cells. Their function is to transport oxygen from the lungs to the rest of the cells of your body, and then to smaller degree, transport carbon dioxide back to the lungs so that you can exhale it. These are considered not to be true cells. The reason for that is they are anucleated. They do not have a nucleus, okay? So this is one reason that they're considered to not be true cells. So just a little factoid for you. Leukocytes, these are your white blood cells. There are many different types of white blood cells. These are your second line of defense. Does anybody know what your first line of defense is?
Your skin.
That's right. Excellent. Your skin is the first line of defense. Then you have your white blood cells, your leukocytes being your second line of defense. Then our thrombocytes or platelets. I just didn't know thrombocytes. I always use the word platelets. Okay, these are really important in blood clotting. And we kind of talked about, no, that was physiology class, sorry. I'm going to get myself confused about which class I talked to about something, so I apologize now. But they are involved in blood clotting, and let's say you get a cut, and maybe you cut yourself with a kitchen knife, and it looks like a nice smooth cut, but microscopically, you've got rough edges on that cut. And those platelets are going to burst open when they flow past those rough edges, and they release chemical signals that are like SOS signals saying, help, we need to clot this blood, we need to stop this bleeding. And it starts this whole cascade of reactions for blood clotting. So platelets have a really important role in the blood clotting process, not only in releasing chemical signals, but also kind of helping to form the clot itself. Okay, this is a little fuzzy, or it's my eyes, but this is just kind of giving you a quick rundown of the different types of blood cells. So our red blood cells, or our erythrocytes, they're kind of like a donut, except for it has like a squeezed in the middle. Like, let's say you got a jelly donut and just squeeze the middle in. So you don't have a hole there, but there's like a membrane kind of in the middle, but then you have the rest of your donut. And that's kind of how red blood cells are shaped. And they're shaped that way. It helps increase the amount of surface area on each of those red blood cells to be shaped that way, which allows them to carry more hemoglobin, which allows them to carry more oxygen. So you're maximizing how much oxygen you can carry by having that specific shape. White blood cells, not going to go into too much detail with these guys. You have monocytes, lymphocytes, eosinophils, neutrophils, and basophils. So monocytes, these are phagocytes, and you'll find them in other tissues, kind of like macrophages. They wander around looking for things to eat, looking for things that don't belong. Lymphocytes you're going to find in the lymphatic system, specifically in the lymph nodes. They are there screening the lymph fluid that's coming through the lymph node. And if there's any infection, bacteria, viral infection, they're going to mount an attack and fight that infection. And that's when your lymph nodes get swollen and sore, is when you're having an active infection being fought in your lymph nodes via the help of lymphocytes. Let's see. Eosinophils, neutrophils, and basophils all are involved in the inflammatory process. That's really all we're going to go to. We're not going to go that far on these guys. So they're all involved with inflammation. For example, eosinophils are involved with launching an attack towards parasites, like a parasitic infection. Basophils and mast cells have histamine that they release. And oh my goodness, I've forgot what neutrophils do. Oh, well, it's all right. Neutrophils are phagocytes. There we go. Phagocytes. So there are also phagocytes like monocytes and macrophages. And we've talked about platelets. They're like little membrane enclosed sacs of chemicals. And they are important in blood clotting. Okay, bone. This is that solid connective tissue. So we're looking at, this picture is of compact bone. So the cell type is an osteocyte. So looking at this picture, these little dark areas is where you would find an osteocyte. Osteocyte lives in, it's like a little cave, lives in that little cave called a lacunae. And then I'm sorry, I keep wanting to get really detailed, and I know we don't need to. But this is a central canal right here where you have your blood supply that come through each of these round osteons. Okay, so compact bone, you have lots of osteons. In the central canal, or the Haversian canal, is where blood vessels are. And you have these little tiny canals that go to all these cells trapped in the lacunae to give them the nourishment, to help take care of them. So this is in the skeletal system, and it's involved with calcium storage, and it can also be, it is calcium storage. For example, if you don't have enough calcium in your blood, your body basically tells, well, hormones tell your body to release some of the calcium from your bone. So it is considered a calcium storage. So it's really important to make sure you are getting enough calcium in your diet, because if you're not, you're going to be leaching calcium out of your bones and not putting enough back. And that's going to lead to things like osteoporosis and bones that are easier to break, and you don't want to have that problem. And unfortunately, ladies, we are more susceptible to this, so we got to get our calcium and our vitamin D every day. If you take calcium but don't take vitamin D, it won't work. You can't absorb it without having vitamin D. Okay. So with bone, the main unit is called an osteon. So it looks like a tree with rings around it. That is our osteon. There's our central canal right here. Our osteocytes stuck in their little lacunae caves. And then those little lines that I was pointing out earlier, those are called caniculi. Caniculi literally mean little canals. And so they are carrying the nourishment, the blood, from the central canal out to all of these osteocytes that are stuck in these lacuna caves. Because these bone cells are not super smart. They build bone around them until they've trapped themselves in bone. And so then they have to rely on these little caniculi to bring the nourishment from the bloodstream to each individual cell. So skeletal, I almost said skeletal muscle. The skeleton is very vascularized. There's a good bloodstream, good blood supply to the skeletal system. Okay, so osteocytes, bone cells, oh, we just talked about this. We're just gonna, oh, lamellae. Lamellae would just be the solid portions here, for example, like the rings of a tree, that would be a lamellae. Okay, cartilage. This, I love this type of cartilage. I think it's pretty. This is hyaline cartilage. And you typically find small little groups of chondrocytes, which are the cells here. And everything around that is the matrix that's been secreted by the chondrocytes. So let's see, where do you find cartilage? You find it between each of your vertebrae, between joints, within your ribs, and connecting your ribs to your sternum, for example. So functions, oh, also in your nose, that's elastic cartilage, just so you know, in your ears. Reduces friction within joints, provides flexibility and structure and strength. Okay, hyaline cartilage, you find these in joint cavities and between the ribs and the sternum. So let's see if I can stand up a little bit. So you have your ribs right here. This is actually cartilage. Your ribs stop over here, and then you have a cartilage web that basically comes up to your sternum where it connects. So that is one of the places that you find hyaline cartilage. Okay, elastic cartilage, couple places you find that. Your ear, so it bounces back. It's very flexible. The epiglottis down in your larynx is also made of elastic cartilage. Let's see, any place else? So this type of cartilage allows a lot of movement, distortion, and the ability to kind of basically bounce back to its original shape, which is really cool. Story time. My brother-in-law did this to his teachers when he was in elementary or junior high. He spent his whole PE period bending his nose to one side. Well, if you hold it to one side long enough, it will stay for a while. So he did this, and then he went up to his teacher and said, "I think I broke my nose," and freaked the teacher out because his nose was bent. That's made out of cartilage, by the way, and it has the ability to go back to its normal place. But if you hold it in position long enough, it will stay there for a little bit. And he really scared his teacher. And it's kind of, I think it's a funny story. Probably shouldn't do it to your teachers. Or do it to your friends, scare them good. Tell them your anatomy teacher told you. Okay, let's see. Fibrocartilage. You find fibrocartilage in between the discs, or they make up the intervertebral discs between the bodies of the vertebra in your spine. So they're the padding you see here in blue. Those are your intravertebral discs. This is the strongest type of cartilage, and there are also fibers in it. So when you look at this, you'll see the typical kind of cartilage cell type, but you'll also see fibers. That's the only type of cartilage that has fibers in it. So that's kind of an easy way to remember how to identify this, is you look for those kind of round, blobby cartilage cells that are in small groups, and then for the fibers that surround them, that's going to be fibrocartilage. Other places you'll find fibrocartilage is between the pubic bones in the pelvis. There's a joint called the pubic symphysis, and that is held together by fibrocartilage. Padding in your knee joints, so your meniscus, is essentially fibrocartilage. It's thick. It's there for protection and padding to offer some shock absorption. Prevents bone-on-bone contact. You do not want that. Oh, I can't read what that says. Oh, okay, cell terminology. Okay, hypertrophy. This is when cells increase in size. So this can happen with skeletal muscle. So think about somebody who's been working out a lot and they're trying to beef themselves up. That's an example of hypertrophy. Hyperplasia, on the other hand, is when you have an increase in cell number. Often, I'll back down. Okay, so early stages of pregnancy would be a good example of hyperplasia. You can also see hyperplasia in early stages of like precancerous growths where you have an increase in cell number. They'll call it hyperplasia. It's not cancer, but it's made some changes towards that direction when it's hyperplasia. Metaplasia is when you have a change from one cell type to another. Okay, so for example, when someone is a smoker, in their respiratory system, the simple columnar cells actually change to squamous cells instead. And that is a physical change that occurs in somebody who is a regular smoker. So here's just kind of an illustration showing you the difference between hyperplasia and hypertrophy. So if we start out with 4 cells, hypertrophy is we still have 4 cells, they're just bigger. So the cells you haven't increased the cell number. So with skeletal muscle, you're not increasing the skeletal muscle cells, you're just increasing the size of them, okay? With hyperplasia, on the other hand, you're increasing the cell number. And that can be great in the early stages of pregnancy, but can be bad if your body has made some changes and you're starting to create a growth that you don't want. Okay, and then here, oh, I forgot, is metaplasia, where you have columnar cells changing into squamous cells. And so this is something that we know does happen in smokers. So to squamous metaplasia. Okay, intercellular connections. We're almost done with this chapter. Oh my gosh. Guys, can you hang on for like 2 minutes? Okay. We're almost done. Okay, I'll go quick. So there are three types of intracellular connections. There are desmosomes, tight junctions, and gap junctions. So desmosomes, they're very abundant and they connect to the cell basement membrane. So they're found at the bottom of the cell walls. Tight junctions are interlocking membranes, and they form basically like I can't think of the word for it, but they basically are impermeable. So nothing can pass between those two cell walls because you have those tight junctions. And gap junctions actually have a pore that connect cell to cell so that the cells can communicate with each other, allows the movement of ions, which allows communication. So I put this picture up so you guys could see the different types of junctions. Here are gap junctions, and there are little pores you can see over here in the gap junction that allow ions or small molecules to pass back and forth. Tight junctions prevent substances from being able to move down between those cells. So it makes it like it's glued closed. Nothing can pass through. And then you have desmosomes, which are basically, there's fibers that hold these cell walls together, and they're anchoring. Oh my gosh, I thought we were done. Okay, I'm gonna stop because I know we're out of time. We're seriously almost done with this lecture. We're gonna get through the skull and into the ears and eyes with our next lecture, okay? So be prepared for that. We are going to rock this. Have a great night. Thank you so much for coming to class. If I did not call your name, please don't go until you've given me your attendance, okay? Thank you so much. Have a great night. Oops. Yes, I want to keep. Okay. So did Madeline Johnson show up today? Jessica Lucero?
Right here.
Ah, all right, good. Check you off. And then Olivia Rogers. Maisie Shalau. Did I hear something? No? Okay, awesome. Thank you so much, guys. I will make sure there's another link up before our next class, and keep doing your great work.
Megan is here. She doesn't have audio.
Megan. What's Megan's last name? Anybody know?
Madeline Johnson.
Oh, okay, perfect. I got her. Thank you. So we only have 2 people that we missed today. That's really good. Thank you all so much for showing up. Now,
Did you get me by chance?
okay, who's talking?
Macy Lawler.
Macy Lawler, let me see.
I was a little late.
Yes, I did get you.
Okay.
Yes. Anybody else need to check?
I got more of a question.
Sure.
If we can get out a full screen here.
Okay, let me figure out how to do that. Oh, hold on.
Yeah, you want to save that?
Yeah, I do want to save that. I need to figure out, okay, here we go. Almost a full screen. I'm screen sharing. I can stop that. Okay, we're at full screen. Okay.
Okay. So I just want to make sure I know where the quiz is.
It is on Canvas.
Yep, I'm in Canvas. Where should I, is it in the courses or?
It is in the quiz section.
Okay.
Is there on the drop down menu on the left hand side, should be a bunch of things listed. There should be one that says quizzes.
Not seeing it.
You're not seeing it. So
I see home announcements, syllabus, modules, grades, people, my media.
oh, check assignments.
I found it in the syllabus tab.
You found it in the syllabus tab?
Yeah.
Oh, yeah, I do see it down there. Okay.
Oh, there it is.
That's a great place to find it. Thank you. Perfect. Yeah, I'm
Okay.
still working on getting everything finalized on Canvas. So there are a few things that are still a little messed up.
Okay.
All right. Any other questions?
Did you get me, Elizabeth Morales?
Let me check. Yes, I did.
All right, thank you.
Sure thing. The only person, there's two people I did not get. Olivia Rogers and Maisie Shalau. So everybody else, you've been checked for today.
I have a question.
Yeah.
So we have the quiz due tonight, right? And then our homework's due on Wednesdays. Is that right?
Checking your next homework will be due, well, you have homework due tonight, but then your next homework is not due until September 9. So you have,
Okay.
you have some time.
Okay.
I posted up a separate lecture schedule, just a PDF, so you guys could look at just the schedule if you wanted to. So this will be really helpful for you. If you see there, it should be really helpful for you as you're kind of looking at stuff. And you should be able to find it in the module section on Canvas. But if you have trouble, let me know and I can send an announcement out with the PDF attached to it.
Okay. Yeah, I found it.
Okay, cool.
Okay.
Wait, is the homework on Top Cat or is it in Canvas?
Top Hat.
Homework's on Top Hat.
Okay,
Canvas
okay.
is
I
gonna
think,
be quizzes and exams.
okay, I'll go double check then.
Okay.
Because my Top Cat wasn't telling me when it was due. I was just kind of estimating, I guess, on which ones were due.
Okay.
On the top hat.
So
on
I
the
was kind of
assignments,
confused.
You
I was
put
trying
midnight for the closing, so it closed at midnight this morning, not 11:59 tonight.
Oh,
So
really?
We're
Yeah.
already closed out.
Oh, let me fix that right now.
Yeah, I saw that
That's good to
yesterday.
know.
I
saw that-
thought I had it set the right way. I can fix that.
Yeah.
So
I think you
Oh.
have to
Set
put it
it
to
for last night.
11:59 instead of the 12 a.m.
11:59. That's kind of funny. I'm sorry.
And then the other thing, I had a question on the worksheet too.
Yeah.
Like my answers are right, but they didn't match your answers word for word, so it marked them as wrong.
I have to
Correct.
go through them and
Mine have done the same thing for both quizzes.
I have to go through them and recheck everything, unfortunately.
Okay.
So don't
I just
worry
wanted to
about
make
it
sure
yet.
that was something you were planning to do.
I don't want to do it, but I have to do it. It's a lot of work, but yeah. So let me see if I can fix like worksheet 1 really quick and get it set right. So if
Yeah,
I
because I saw that
October-
yesterday. I was
Oh,
like, oh crap, it was due last night at midnight, so I quick did it yesterday afternoon.
oh. So let me put 11:59 p.m. Okay, so somebody check for worksheet 1.1. I just changed that. So it should be available now again.
To redo,
Yeah, it's available again.
Okay, so I will fix that for the other assignment. I'm fixing it right
Is-
now.
is that to redo or?
That's if you haven't done it yet, because it was, somebody get down.
Well, worksheet 1, okay, so like I put the left hyper.
You put what?
Oh,
There.
she lost her connection.
Oh shoot, lost ya. Maybe she'll come back.
I have a question about the syllabus.
Okay, yes.
When it says quiz, does that mean what's above it?
Yes. So
So
it just means there's a quiz that day, and the quiz is on the stuff that we covered in class already. And
Okay.
then it gives a topic of what we're going to talk about in class that day.
Okay. Okay, cool.
Yeah, I'm sorry. I know that's not
I think
super
my-
clear.
My top hat just. I think my top hat just kicked the classroom.
I'm sorry, you broke up there. Can you say it again?
I think my top hat, it kicked me out of the classroom.
Oh, it did?
Yeah, because well, now it's being.
Well,
let's see,
if you can't get in, send me an email, because if I have your email address, I can go in and re-invite you.
Sorry, my computer died.
Okay. Is it 1.1 you said?
I had on the homework assignment like the left hypochondriac region, and it was just supposed to be left hypochondriac.
That's something
I even went into the tutor, and there was like 4 other questions that were on that that I had gotten right, but because I had added words or because I was trying to get through on the computer and it was misspelled, it was counted wrong.
I will go back through and check everybody's answers and fix those.
Okay.
So, I mean, if it's obvious that the answer is what the answer is, I'm going to give you the points for it.
Okay.
Okay.
That's all I was wondering.
Yeah. So I don't know.
I just didn't know if like Top Hat was computer generated and that's why they're coming up wrong.
Yes.
Okay.
So because there are the chances for errors, I have to go back in and double check everything.
Okay.
Yeah. So give me a couple days to do that with the first assignment. It'll probably take me a couple days to get through those, but I will do it.
Okay. So the assignment that was due last night, there isn't one due tonight? Because I can't find anything due tonight.
I just switched it. It was accidentally midnight last night instead of midnight tonight. So I just fixed it so it's due tonight. So if you want to go back through and look at things, you can. But there is nothing new due.
Okay.
So just your quiz. If you've already done your quiz, you're good to go. And that's on Canvas.
Okay.
Your homework.
Okay.
If you've already done that, then you're good. And I will check everybody's homework questions.
All right. Thank you.
Sure. Any other questions?
I do. Is it supposed to say the correct and wrong answer on there, or because I put it in, I put submit, but it doesn't show if it's right or wrong?
On Top Hat?
I did both of them.
I don't know. Top Hat is new to me, just to be honest.
Okay.
It doesn't show the answers until after the time of it's closed, and then you can go back and hit review and compare your answer to the answer that is put in the system.
Thank you.
Okay.
So since she reopened it, we can't see the answers anymore.
Oh,
but when
sorry. I reopened. Some people still hadn't done theirs, and I was trying to be nice. So you'll be able to review your answers tomorrow, or tomorrow morning, or after midnight tonight. Okay.
All right. Thank you.
Yeah. Anything else?
I think we're good, so
Okay.
hope you feel better.
Thank you. I am feeling okay. I have to go get COVID tested tomorrow, which I'm not excited about, but we got to do it. Just to
Sorry, guys. I was kind of dealing with baby tonight.
Oh my gosh. You have a deer.
Yeah.
It's a baby deer, yes.
That's
but
awesome.
I was trying to like pay attention, write stuff down, and then she came up and she's like, I'm hungry.
Yeah. Our animal friends don't get it. That's really sweet. That's neat. I can't hear her.
Oh, she said that's her baby.
Oh, that's your baby. Okay.
Yeah, her baby. Yep.
That's cool. Very cool. All right. Well, I'm going to let you guys go.
Sounds
I'm
good.
gonna start recording this lecture and try to get it on YouTube for you.
Sounds good.
All right.
Sounds great.
Have a great night.
You too.
Thank you. Stay
Thank
safe.
You too. Bye.
Bye.